Brain-breathing connection. A potential breakthrough in hypertension treatment, new research suggests that the key to treating high blood pressure may not lie in the heart or the arteries, but in a specific region of the brain responsible for controlling breathing. A collaborative study by the University of São Paulo and the University of Auckland has identified a potential biological driver for hypertension: the lateral parafacial (PFL) region. This discovery could offer a lifeline to the millions of people whose blood pressure remains uncontrolled, even with standard medication. The breathing blood pressure link, the PFL region, is primarily known for its role in managing forceful, deliberate exhalations, such as those occurring during heavy exercise, coughing, or laughing. However, researchers have discovered that this area does much more than regulate breath. In laboratory tests involving rats, scientists found that the PFL region acts as a bridge between respiratory rhythms and the sympathetic nervous system, the body's fight-or-flight response. When these neurons are activated, they trigger a cascade of signals that cause blood vessels to constrict, leading to a spike in blood pressure. This mechanism helps explain several medical phenomena. Uncontrolled hypertension. Approximately 40% of patients fail to reach target blood pressure levels despite taking medication. This suggests a neurogenic cause where the brain is essentially driving the pressure up from the top down. Sleep apnea risks. People with sleep apnea often suffer from high blood pressure because their breathing is interrupted during sleep. Low oxygen and high CO2 levels trigger the PFL neurons, inadvertently activating the body's blood pressure-raising mechanisms throughout the night. From discovery to potential cure, the most significant aspect of this study is not just identifying the cause, but demonstrating that it might be reversible. Using genetic engineering, the research team was able to manipulate PFL neurons in animal models. They observed that activating these neurons triggered brain circuits that raised blood pressure. Inactivating these neurons caused blood pressure to drop back to normal levels. When our team inactivated this region, blood pressure fell to normal levels, noted physiologist Julian Patton of the University of Auckland. The next frontier, targeting the brain without entering it while the findings are promising. A major hurdle in neurology is the blood-brain barrier, the protective layer that makes it incredibly difficult for drugs to reach specific brain regions safely. To bypass this, researchers are looking at a remote control approach. They are focusing on the carotid bodies, clusters of sensory cells located in the neck. These sensors detect changes in blood chemistry and send signals to the PFL region. The goal is to develop a drug that targets these neck sensors instead of the brain itself. By quenching the activity of the carotid bodies, scientists hope to indirectly quiet the PFL region, lowering blood pressure without the risks associated with drugs that must penetrate the brain. Looking ahead, it is important to note that this research has currently only been conducted on animal models. While it is highly probable that similar circuitry exists in humans, extensive clinical testing is required to confirm these results and ensure safety. Given that hypertension affects roughly 1/3 of the global population and is a leading driver of heart disease and dementia, finding a way to target the nervous system could revolutionize how we manage cardiovascular health. Conclusion: by identifying the PFL brain region as a driver of hypertension, scientists have opened a new door for treatment that moves beyond traditional heart medication toward targeting the neurological roots of blood pressure regulation.